Hi, my dear students. This is your Prasanna Harikrishna sir from SRR Government Arts and Science College, Karimnagar. So today we are going to see the oxidative phosphorylation of respiration. Okay. So there is important process which can be seen in electron transport system of respiratory mechanism, that is aerobic respiratory mechanism. Clear? So what is meant by oxidative phosphorylation? Clear? So there are different kinds of phosphorylations. So in this context we are going to see and discuss about oxidative phosphorylation. Remember my dear students, phosphorylation, whenever you hear the term phosphorylation, it indicates addition of phosphate. Addition of phosphate. So addition of phosphate to any compound. For example, if you add phosphate to glucose, so glucose plus phosphate, if you add one phosphate to glucose, it will become into glucose monophosphate. In glycolysis, you can say if one phosphate is added or attached at the sixth carbon of glucose, that can be called as glucose 6-phosphate. Clear? In the general context, if one phosphate is added to the glucose, this glucose can be converted into glucose monophosphate. Mono nothing but single. One single phosphate is added to the glucose structure. Clear? Or if one phosphate is added with ADP, that is adenosine diphosphate. Okay. So one phosphate is attached or added to ADP. This can be converted into one ATP molecule, that is adenosine triphosphate, that is called as energy molecule, or you can say cell currency of living organisms. Clear? So ATP is formed, adenosine triphosphate is formed whenever the phosphate and ADP combines together. Clear? So adenosine diphosphate and one particular phosphate molecule. Okay. So this combination will lead to the formation of one molecule of ATP. Clear? So this is called as what? Phosphorylation. Simply you can just remember phosphorylation nothing but what? Addition of phosphate to any of the compounds. Okay, so any of the compound means in this context addition of phosphate is taken place with glucose. So hence there is a formation of glucose monophosphate. So in this context one phosphate is added to the ADP. Then there will be a formation of one molecule, that is ATP, adenosine triphosphate. Okay, but remember in front of phosphorylation there is one more word, that is oxidative. So oxidative nothing but what? Addition of oxygen. Oxidation means what? Addition of oxygen. Remember. So this is what already in the last class I have drawn this entire picture. That is how the electrons move from complex to different complexes. Okay, there is a movement of electrons from complex 1 to complex 3, complex 2 to complex 3, and complex 3 to complex 4. It's clear? So I have already explained all these complete information. So now in this session, so this particular electron movement accompanied by the formation of water at the last complex, that is complex 4. Okay, so these electrons are received by oxygen and protons, hence there is a formation of water. And so whenever oxygen is available, there is a formation of water. There is a combination of oxygen with protons and electrons, and formation of water will be taken place. Clear? So whenever the oxygen is available, there will be a formation of one molecule of ATP. Remember. Okay, so here there is a formation of ATP, that is adenosine triphosphate. This ATP will be formed whenever the oxygen is available. So ATP is formed in the presence of oxygen. So if there is no oxygen, there is no formation of ATP. Clear? So in the presence of oxygen, there will be formation of ATP. Hence this entire process, this entire mechanism can be called as oxidative phosphorylation. Okay, so now the formation of ATP, is based on the different biochemical processes. So this entire formation of ATP, that is phosphorylation, oxidative phosphorylation, can be explained by the Peter Mitchell. Remember, Peter Mitchell. So Peter Mitchell proposed a theory called chemiosmotic hypothesis. Chemiosmotic hypothesis. So chemiosmotic hypothesis proposed by Peter Mitchell in the year 1961. For this, this is beautiful process or beautiful phenomenon. So for this proposal, for this description, he got Nobel Prize in the year 1976. Clear? So this is about Peter Mitchell's chemiosmotic theory. What is chemiosmotic theory? So what kind of information the chemiosmotic theory gives us? Remember, chemi nothing but chemical, osmotic. Osmosis means what? Osmosis means what? So movement of protons from lower concentration to higher concentration, or there is a diffusion process also. So osmosis and diffusion. Okay, there are completely two biochemical processes. So in this context, so chemiosmotic hypothesis, how it explains about the formation of ATP from the ADP and phosphates. Just look at the board. So for every electrons, for the movement of electrons from different complexes, so from complex 1 to ubiquinone, there is movement of 4 protons from the matrix to perimitochondrial space. Then complex 2 to complex 3 means complex 2 to ubiquinone, there is another 4 electrons which move from matrix to perimitochondria. Then there is whenever this electrons reach complex 4, the 4 protons from matrix gets moved from matrix to perimitochondrial space. But so there is a small change in this particular moment. 2 protons are utilized for the formation of water, remaining 2 protons moved from matrix to perimitochondrial space. Clear? The total number of protons, if one NADH2 of Krebs cycle and link reaction. Okay, so one NADH2 of Krebs cycle or link reaction, it is generally participated at complex 1. Okay, so if NADH2 of Krebs cycle and link reaction participate at complex 1, how many protons from here to there? Just calculate how many protons move from matrix to perimitochondrial space. So 4 plus 2. Okay, so 1 NADH2. So I'll write here 1 NADH2 of Krebs cycle and link reaction. Okay, equivalent to 4 plus 2 protons means 10 protons. Clear? Now, so this is what FADH2 of Krebs cycle, this is what NADH2 of Krebs cycle. Okay, so both FADH2 and NADH2 of glycolysis do participate at complex 2. Okay, now you can count from here to there only. You can skip the complex 1, that is 4 protons, because so FADH2 of Krebs cycle, NADH2 of glycolysis do participate at the complex 2 only. That's the reason, that's why you can count the proton number from here to here only. Okay, so now 1 NADH2 of glycolysis, next FADH2 of 1 FADH2 of Krebs cycle. So will yield us 6 protons and 6 protons. So you can count 4 plus 2, 6. Okay, so 1 NADH2 of glycolysis is equals to 6 protons. 1 FADH2 of Krebs cycle is equals to 6 protons. Clear? Now, so remember, so now, whenever one NADH2 participates at complex 1, it will give rise to 10 protons. Then tell me, what about the total number of NADH2s which are formed in the aerobic respiratory mechanism? Just remember, so In glycolysis, right, in glycolysis, the total number of NADH2s in glycolysis. So this is glycolysis. The total number of NADH2s are 2 NADH2. So 2 NADH2s are formed in the glycolytic pathway. So glycolytic NADH2 is equals 1 NADH2 of glycolysis will give rise to 6 protons only. Then here only here 2 NADH2s are formed is equals to 6 plus 6. 6 plus 6 is equals to 12 protons. Okay. Then come to link reaction. In link reaction, 2 NADH2s are formed. So link reaction NADH2 is equals to 10 protons. Hence, 10 plus 10 is equals to 20 protons. Clear? Now, the Krebs cycle. So Krebs cycle, there are, if once acetyl CoA enters into Krebs cycle, there will be formation of 3 NADH2. But 2 acetyl CoAs are formed in the link reaction. Hence, 3 plus 3 is equals to 6 NADH2. Okay, 6 NADH2. I have already told you the complete information in the Krebs cycle. Just you can go through that video. Okay. So 6 NADH2 is equals to how many protons? Okay, 1 NADH2 is equals to 10 protons, but here 6 NADH2 will give rise to 60 protons. 60 protons. Then in Krebs cycle, 1 FADH2, if acetyl CoA enters into Krebs cycle, 1 acetyl CoA is equals to 1 FADH2. 2 acetyl CoAs enter into Krebs cycle, there will be formation of 2 FADH2, right? So now 1 FADH2 is equals to 6 protons, but here 2 FADH2s are formed in the Krebs cycle. The total number of protons will be 12 protons. Okay, clear? One glucose molecule, if one glucose, they are equivalent to, the total number of protons are equivalent to 1 glucose molecule. If one glucose molecule enters into aerobic respiration into different stages, that is glycolysis, link reaction and Krebs cycle, there will be formation of 2 NADH2, 2 NADH2 in link reaction, 6 NADH2 in Krebs cycle, 2 FADH2 in Krebs cycle. So all the NADH2 and all the FADH2 will give rise the total number of protons. These total number of protons equivalent to 1 glucose molecule. Okay. Now just imagine if 1 glucose molecule enters into aerobic respiratory mechanism, the total number of protons will be formed, will be moved from matrix to perimitochondrial space. Okay, just count them: 12, 20, 32, then 92, 94, 104. So total 104 protons move from matrix to perimitochondrial space. Just imagine how the mechanism occurs inside the mitochondria. Remember, okay? So this 104 protons for each 1 glucose molecule move from matrix to perimitochondrial space. What happens now? Now what happens? So there is a deficiency of protons in the matrix. There is increase in the proton gradients in the perimitochondrial space. Gradually proton number increases in the perimitochondrial space. That is called as what? Gradients. So proton gradients is increased in the perimitochondrial space. Proton number gradually decreases in the matrix of mitochondria. Clear? Now what happens after reaching the peak levels of proton number in the perimitochondrial space? So now what happens? Every 3 protons. So this is what? F0. This is F1, F0, F1 complex, or you can say oxisome, or you can say Fernandez boron particles, or Parsons particle, or elementary particles, or primary particles. This is the smallest rotating part in the globe or in the universe. Clear? So these are the names, or ATP synthase or ATP synthetase, you can say. Clear? So this complex phi, or you can say F0, F1 complex or oxisome, involves in the formation of ATP. So for ATP formation, so here 104 protons, the total number of protons for one glucose molecule is 104. Okay, out of 104, only this F0 channel, this is a channel, this channel will allow only 3 protons at once. Okay, for example, there is a door, there is a door to every room. So door does not allow 3 or 4 members at the same time. Okay, so door will allow, or entry or exit, it allows 1 or 2 persons at the single time, at the same time. Clear? In the same manner, this particular channel, this particular path, passage will not allow more than 3 protons. Clear? So it allows only 3 protons at once. Clear? So for every 3 protons, right, these 3 protons move from the oxysome. So whenever they get moved from oxysome to where? Matrix, means perimitochondrial space to matrix. Right? So there is a head portion of oxygen. This is a complex 5. Head is there now. Head is located towards the matrix of mitochondria. Clear? So whenever then the 3 protons moved from the perimitochondrial space to matrix, there is a small pressure. There is a small pressure. So due to that particular pressure, this particular head region, or a pin-like region rotates like this. Whenever it rotates, this ADP combines with phosphate and there will be formation of one single ATP. Okay, for every 3 protons, from perimitochondrial space to matrix, there is a formation of one ATP. So this is what oxidative phosphorylation according to Peter Mitchell. This is chemiosmotic hypothesis. So chemi, nothing but protons. So due to osmotic pressure, low higher osmotic pressure is there in the perimitochondrial space, lower osmotic pressure is there in the matrix of mitochondria. So due to the difference in the gradients of protons, these protons move from perimitochondrial space to matrix of mitochondria. So for this movement, this layer cannot allow the protons from perimitochondrial space to matrix, but only complex 5 allows the protons from perimitochondrial space to matrix. But conditions apply, only 3 protons at once. 3 protons move through F0 and F1 to the matrix of mitochondria. For every 3 protons, one ADP combines with phosphate and there is a formation of ATP. Okay, now just analyze the information. So for one NADH2 of Krebs cycle, one NADH2 of Krebs cycle, this is what? Krebs cycle. So I will rub it. Right, see, one NADH2, this is what? Krebs cycle. Okay, so one NADH2 of Krebs cycle is equivalent to how many protons? 10 protons. Right, 10 protons. Right, for every 3 protons, there is a formation of one ATP. So then what about 10 protons? 10 protons are equivalent to how many ATPs? Approximately 3 ATPs. Okay, let it 1. Okay, so 10 protons are equivalent to 3 ATPs. Then one NADH2 of glycolysis. So one NADH2 of glycolysis is equivalent to 6 protons. So 6 protons, hence there is formation of 2 ATPs. Then come to one FADH2 of Krebs cycle. So here also equivalent to 6 protons, hence there is a formation of 2 ATPs. Clear? So now the total number of NADH2 which are formed during the Krebs cycle, or the total number of Krebs cycle NADH2s are 6. One NADH2 is equals to 3 protons. Right? One NADH2 of Krebs cycle is equals to 3 ATPs. but the total number of NADH2 which are formed in the Krebs cycle are 6. Okay. So 1 is equals to 3 ATPs. 1 NADH2 is equals to 3 ATPs. Then what about 6 NADH2? 6 NADH2. 1 is equals to 3. 6 is equals to how many? Simple. So 6 into 3 is equals to 18 ATPs. Okay. The total number of ATPs are 18. Clear? Then come to 1 NADH2 of link reaction. Then link reaction like Krebs cycle and link reaction also. Krebs cycle and link also equivalent to how many? 10 protons. I have already told you in the previous minutes. So Krebs cycle and link reaction will give rise to 10 protons. 10 protons is equivalent to the formation of 3 ATPs. Clear? Then come to link reaction. So link reaction also 3 ATPs. Then how many NADH2s are formed in the link reaction? 2 NADH2. Right? So 3 into 2 into 3. 2 into 3, that is 6 ATPs. Clear? Then come to 1 NADH2 of glycolysis. So 1 NADH2 of glycolysis is equivalent to 6 protons, right? This is glycolysis, 6 protons. 6 protons are equivalent to how many ATPs? 2 ATPs, right? So 2 ATPs. Then tell me, how many NADH2s are formed in the glycolysis? So how many NADH2s are formed? 2 NADH2s. So 2 into 2 is equals to 4 ATPs. How many? 4 ATPs. Then come to 1 FADH2. So 1 FADH2 of Krebs cycle. So 1 FADH of Krebs cycle gives rise to 2 ATPs. Then how many FADH2s are formed in the Krebs cycle? 2. So then 2 into 2 is equals to 4 ATPs. Okay. Then count them all. 18 plus 6. 24, 24 plus 4, 28, 28 plus 4, how many? 32. Right? 32 ATPs are formed in, the entire, so 18 plus 6, 24 plus 4, 28 plus 4, 32. So 32 ATPs are formed indirectly. Okay. 32 ATPs are formed indirectly in all the stages of aerobic respiration. Then what about direct formation of ATP? So I have already told you in the glycolysis and Krebs cycle. In the glycolysis as well as in the Krebs cycle. Clear? So now the direct formed ATPs, right? So I am writing here direct ATPs in glycolysis. Directly formed ATPs in the glycolysis. So from one side 2 and another side 2 means the total 2 plus 2 is equals to 4. Okay. Then direct ATPs formation in Krebs cycle. So Krebs cycle, there is, if one acetyl CoA enters into one Krebs cycle, there is a formation of 1 ATP. So acetyl, there are 2 acetyl CoAs are formed. Then 2, if the 2 acetyl CoAs are involved in the Krebs cycle, there will be formation of 2 ATPs. So 1 plus 1 is equals to 2. Then count them all. 32 plus 4, 36 plus 2, 38. Clear? The total number of ATPs. The total number of ATPs which are formed in the aerobic respiration. Clear? The total number of ATPs, so which are formed in aerobic respiration are 38. Remember, 38. Okay. So out of 38, 2 ATPs are utilized. So 2 ATPs are utilized in the glycolytic pathway at the previous steps. Okay. 2 ATPs are utilized. The total number is 38, 2 are utilized. Then the net ATP number will be 36. Net ATP number will be 36 ATPs. Okay. So out of 36, 1 ATP, for example, 1 ATP molecule will give rise 6.3 kilocalories of energy. Sometimes it is 7.6. So this is 7.3 kilocalories. Sometimes it is, some books, the information related to ATP energy, so maybe 7.6 kilocalories. Okay. So if you calculate with 7.3 kilocalories, if 1 ATP is equals to 7.3 kilocalories, what about 36 ATPs? So 36 ATPs is equals to 36 into 7.3 kilocalories. The total number will be, just count them. Just count them. The total number, just you can calculate. Okay. So this is about, the entire picture of chemiosmotic hypothesis or oxidative phosphorylation. Okay. So this is about, aerobic respiratory mechanism. In next session, we will move to anaerobic respiration, right? So thank you, my dear students. Thank you.